Welcome students to the lecture of Immunology. We shall be studying chapter 3, Organs of the Immune System, where we will discuss about the secondary lymphoid organs, the lymph node. After this lecture, the student should be able to describe the structure of the lymph node, how lymphocytes are activated in the lymph node, the structure of the primary and the secondary lymphoid follicles, and the importance of germinal centers in the secondary lymphoid follicles. So let us start with the lymphoid system. The lymphoid system is basically made up of the immune system and the lymphatic system. The immune system is a system that consists of immune cells that are circulating in the blood and the lymphoid organs. The immune cells are produced in the bone marrow. From the bone marrow, they enter into the blood circulation, and via the circulation, either the blood circulation or the lymph circulation, these blood cells enter into the lymphoid organs. The lymphatic system is a system that consists of lymphatic organs and the clear circulating fluid called as the lymph. The lymphoid organs are classified into two types: the primary lymphoid organs and the secondary lymphoid organs. The primary lymphoid organs are also referred to as the central lymphoid organs, while the secondary lymphoid organs are referred to as the peripheral lymphoid organs. The primary lymphoid organs are the thymus and the bone marrow, while the secondary lymphoid organs are the lymph node and the spleen. So today we will study about the structure and the function of the lymph node. The lymph node is the first organized structure of the secondary immune system. These lymph nodes are a group of small bean-shaped organs that are present all over the body. Usually, they're present at the sites of lymphatic junctions, right, where the lymphatic capillaries get converted into the lymphatic vessels, and these vessels, before they get converted into the ducts, the junction between the vessels and the trunks and the junction between the capillaries and the vessels are the sites where the lymph nodes are present. There are a number of lymph nodes which are present in the neck region and the trunk. As compared to the other parts of the body, the number is very large in the neck and the trunk. A few lymph nodes are present in the arms and the armpits, as well as the legs of the human and the animals. These lymph nodes are the structures which help to fight against the antigen that is present in the tissue spaces. This is because the lymphatic system carries the lymph from the tissue spaces, and this lymph that is carried by the lymphatic vessels is filtered in the lymph node. So any antigen that is present in the tissue spaces will be carried by the lymph, will be filtered in the lymph node, where in the lymph node the lymphocytes, that is the immunocompetent T and the B cells, will interact with the antigen and help to produce antibodies. Basically, this lymph node is a cellular network of a variety of different cells, including the phagocytic cells and the dendritic cells. So the phagocytic cells, that is the macrophages, and the dendritic cells are the cells that activate the T cells. T cells in turn activate the B cells, and B cells produce the plasma cells producing antibodies against the antigen that has entered into the lymph node. So, a small bean-shaped organ present at various different sites in the body filters antigen from the tissue spaces and leads to the activation of the cells that are present in the lymph node. The lymph node can be divided into three compartments. The outermost compartment is referred to as the cortex. This cortical region is rich in B cells. A very large number of B cells are present in the cortex. Along with the B cells, macrophages, the dendritic cells are also present in the cortex. The dendritic cells that are present in the cortex are present in the primary follicles. The primary follicles are arrangements of B cells or a dense arrangement of a B cell which are naive B cells, that is unprimed B cells that have not come in contact with any antigen. So these cells form a follicle which is referred to as the primary follicle. So within the follicles, along with the B cells, a few dendritic cells are present. And the dendritic cells are present in the follicles and therefore they are referred to as the follicular dendritic cells. Now this cortical region has a very large population of the B cells. But a few T cells may be present here and then in the cortex. And because the large number of B cells are present in the cortex, this cortex is referred to as the thymus independent area. Why thymus independent area? Because T cells are very less, or that you can say there are no T cells in the cortex. T cells are the cells that mature within the thymus. And if T cells are not present, there is no difference made in the structure or the function of the cortex, and therefore this particular area of the lymph node is referred to as the thymus independent region. Below the cortex lies the paracortex, that is the middle portion of the lymph node. The thing here in the orange color is the paracortex. This paracortical region is a region that is rich in T cells. So the population of T cells is much larger in the paracortex, and a few B cells may be present here and there. And because the population of T cells in the paracortex is very large, it is also referred to as the thymus dependent area. Because unless and until the immunocompetent T cells are developed in the thymus, the paracortex cannot function. And therefore, the paracortical area is referred to as the thymus dependent area. Now, as there are some dendritic cells present in the cortex, a few dendritic cells are also present in the paracortex. But now, the dendritic cells that are present are not present in the follicles because there are no such follicles that are present in the paracortical region. And therefore, the dendritic cells that are present in the paracortical region are referred to as the interdigitating dendritic cells because they are present in between the other cells. Okay. The innermost compartment of the lymph node is referred to as the medulla. This medulla contains very few lymphocytes as compared to the number of lymphocytes that are present in the cortex and the paracortex. Right? But the medulla is rich in the antibody secreting cells, that is the plasma cells. And therefore, the lymph that comes out of the lymph node is rich in antibodies that are produced against the antigen that has entered into the lymph node. The lymph node plays a very important function in filtering the antigens and whatever debris are present in the tissue spaces that are carried by the lymph. And it helps in generation of an immune response to the pathogens. In most of the cancer cell patients, the lymph nodes are removed. This is because these tumor cells of the cancer patient may enter into the lymph nodes where within the lymph node these tumor cells can increase in numbers. Okay? That is, metastasized cells that are present that leave the primary tumor enter into the lymph node, and within the lymph node they increase in number, and once the number of tumor cells in the lymph node increases, these cells can pass to the different parts of the body through the lymph. Right? And therefore, in many cancer patients, if you find that the lymph nodes are enlarged, these lymph nodes are removed in order to prevent the spread of cancer to the other organs of the body. We've studied the structure of the thymus, and we saw that the thymus is a bilobed structure that is covered with a capsule. The capsule of the thymus is made up of connective tissue. Right? But the lymph nodes are also structures, bean-shaped structures that are covered with the capsule. But the capsule of the lymph node is made up of collagen, a protein, right, which covers the entire surface of the bean-shaped structure of the lymph node. At a number of places in the capsule, the afferent lymphatic vessels penetrate the capsule and enter into the lymph node. These afferent lymphatic vessels carry the lymph from the different parts and empty the lymph in the lymph nodes. When the lymph comes to the lymph nodes, it contains any antigen if it may be present. Along with the antigen, any lymphocytes that are present in the tissue spaces, the wandering macrophages from the tissue spaces, as well as the dendritic cells from the tissue spaces. So these cells, the lymphocytes, the macrophages, and the dendritic cells are already present in the lymph node, along with which the lymph also carries these cells from the different parts of the body and empties it into the lymph node. So the basic structure of the lymph node can be seen where a bean-shaped structure with a number of afferent lymphatic vessels entering into the lymph node via the capsule. Now the antigen that may be present enters via the lymph into the lymph node. This antigen is carried from the cortex to the pericortical region, the paracortex or the paracortical region. In the paracortex, the dendritic cells and the macrophages that are present, they engulf the antigen, process the antigen, and present the antigen to the T helper cell. This leads to the activation of the T helper cell. We've seen that within the paracortex, the population of the T lymphocytes is very large. So, a few T helper cells get activated in response to the antigen in the paracortex. These activated T helper cells in turn activate the B cells. A few B cells are present here and there within the paracortex. So the B cells that are present in the paracortex get activated, and the activated T helper cell and the activated B cell lead to the formation of small centers at the periphery of the paracortex, right, which are referred to as the foci, because it contains activated T helper cells as well as the dendritic cells and the B cells. These foci that are formed increase in size within 3 to 4 days. After around the maximum size is reached, within 4 to 7 days, a few of the B cells that are activated and the activated T cells leave the foci which are present at the periphery of the paracortical region and enter into the primary lymphoid follicles that are present in the cortex. When these activated T helper cells and B cells enter into the primary follicle, they convert the primary follicle into the secondary follicle. The difference between the primary follicle and the secondary follicle is the primary follicle is made up of B cells that are not activated again with the antigen, that is naive or unprimed B cells. When these activated T and B cells come in contact with the primary follicle differentiates into the secondary lymphoid follicle. This secondary lymphoid follicle shows the arrangement of B cells in concentric circles. Okay. So, the unorganized primary follicle gets converted into an organized secondary follicle, which has the B cells arranged in concentric circles with an area that is present in the center, which is referred to as the germinal center. Right? The germinal center is the site where a large number of proliferating B cells are present. These B cells further get differentiated into the memory cells and the plasma cells. Okay. So the germinal center contains a few proliferating B cells, it contains few memory cells, it contains the plasma cells. The memory cells and plasma cells are the cells that are derived from the activated B cell. In addition to this, we've seen that follicular dendritic cells are present within the germinal centers, and in addition to the dendritic cells, the phagocytic cells, that is macrophages, are also present in the germinal center. So any dead cells that are formed or any B cells that show the formation of a plasma cell which does not produce antibodies against the antigen are killed and are engulfed by the macrophages, and they are removed from the germinal center. Right? So the germinal center is the central position of the center of the secondary lymphoid follicle, in which the activated B cells proliferate and differentiate into the memory cells and the plasma cells. Any B cells that produce antibodies with less specificity towards the antigen are removed by the process of phagocytosis by the macrophages. And the follicular dendritic cells that are present in the follicles keep on activating the B cells so that a large number of plasma cells will be produced, and these plasma cells can produce a large amount of the antibody to fight against the antigen. After the formation of the plasma cells within the germinal center, the plasma cells leave the germinal center, migrate via the paracortex into the medulla. So the medulla is a site where there are a few lymphocytes that are activated lymphocytes, but a large number of plasma cells secreting large amounts of antibodies. The lymph that entered into the lymph node via the afferent lymphatic vessel passes through the paracortex, and finally leaves the lymph node via the medulla. The medulla contains a large number of plasma cells secreting the antibodies, and therefore the lymph that comes out of the lymph node via the efferent lymphatic vessel is very rich in antibodies, as well as is also rich in activated lymphocytes, right? And this lymph is carried from the lymph node and is emptied into the blood vascular system so that the antibodies that are formed in the lymph node can spread throughout the body via the blood circulation. Hope you have understood the structure and the function of the lymph node, how primary follicles get converted into the secondary follicles, and within the follicles, the germinal centers are present where plasma cells are formed. These plasma cells leave the germinal center, come into the medulla, and via the efferent lymphatic vessels, along with the lymph, the antibodies are circulated throughout the body. So the main function of the primary lymphoid organs is to form or to develop mature immunocompetent T and the B cells. But the main function of the secondary lymphoid organ is to provide a site for the interaction between the antigen and the antibody. Antigen and the lymphocytes. Right? So one of the structures of the secondary lymphoid organs is the lymph nodes. These are the references, T.Y.B.Sc and Royd.